Okay, this is key assessment 6C. We're on question 4, plant reproduction. Whee! Barley is an important cereal grown in the UK. It's used widely for animal fodder and a source of maltose. I'm thinking that's probably important when brewing beer. The diagram below is a cross-section through a grain of barley. So what can we see? Endosperm, food store, embryo. Going to give us the seedling. We've got the tester there, so that's the outside of the seed, and then we've got more layers here. So I'm thinking, what's outside of the seed? This will be the ovary wall. Okay. Explain. That means give a reason. Because. Use the word because. Why a barley grain is technically a fruit and not a seed. So, a seed is a fertilised ovule surrounded by a tester. Barley, what are they calling them? Grains. Should we use the technical term? Let's call it an ovary wall. Wall around the tester. So it's a fruit. The endosperm cells contain 21 chromosomes. So what do I know about endosperm? Endosperm is 2 times polar plus a sperm, which is 3n. Calculate how many chromosomes there are in the cells of the embryo. What do I know about the embryo? The embryo is egg plus sperm and is diploid. Explain how you arrived at your answer. So, if 21 is 3n, then n is 21 over 3, which is 7, and we wanted 2n1, 2 7s are 14. So 14 chromosomes in embryo cells. Explanation: endosperm is triploid, so the haploid number is 21 over 3, which equals 7. The embryo is diploid, egg plus sperm, therefore 2 times 7 is 14. That's how I got to my answer. So, onwards and upwards. The table below, and we love a table, shows the results from a study of the germination and early growth of barley grains. So we've got time. The dry mass of the endosperm is going down, and the dry mass of the embryo, so that's endo, the dry mass of the embryo is going up. What can we conclude? What can be concluded from these results? So, the endosperm is being digested, so its mass decreases. barley grain is absorbing products and is increasing the mass. Anything else we can conclude? Right, okay, I don't think it's going to be photosynthesizing because we started off with 42 and we ended up with 34, so it's not yet photosynthesizing. The data is not showing photosynthesis. As the plant is not accumulating biomass. Okay. Oh, this kind of calculation makes me want to cry. Okay. The diagram shows the view from above and from the side of an experiment demonstrating the presence of amylase. So we put our germinating barley grain in. It's producing amylase. It's converting all this to maltose all the way through the depth of the agar. We've got a clear zone, so that's maltose. And we've got bits that are not, which is starch undigested. The agar contained 1 gram of starch per 100 centimetres cubed of agar. Super. The agar in each plate was 5 millimetres deep and the clear zone was 25 millimetres in diameter. Now we've got a problem here because we've got centimetres up here and we've got millimetres down here, which is going to make me cry more. The volume can be calculated. We're given what value of pi to use, so we must use that. Calculate the mass of starch digested. Oh my word. So first of all we're going to need to calculate the volume of the digested area. And first of all I'm going to convert my millimetres into centimetres. So 5 millimetres is 0.5 of a centimetre, and diameter is 25, so the radius is 12.5, but we're in centimetres, so that's 1.25. So let's go for the volume. So volume digested is 3.142 times 1.25 squared times 0.5. Let's do that first. Right, you calculate. Start with your square first if you've not got a squarey button. So if you've just got, the dead cheap calculator like mine. So that's my r squared times 3.142 times 0.5. 2.45. 2.45 centimetres cubed. So we've got 1 gram in every 100. So in every 1 centimetre cubed we'd have 1/100th. 1/100th of starch, grams of starch, multiplied by, we've got 2.45 centimetres cubed, equals 0.0245 grams. Okay, now how many decimal places do they want it to? Show you're working. Give your answer in standard form. So this is the times 10 to the whatever, isn't it? So we're going to start off with 2.45 5, and then we need to work out what we're timesing it by. So timesing by, to take the decimal place to there, times 10 to the, and we're going to the right, so it's 10 to the minus 2. Job's a good one. During germination, it sounds more biology than maths. The amylase is synthesized from amino acids and proteins stored in the barley grain. Name the tissue where the protein is stored, and name the hormone. So, protein, so the tissue is the alurone layer, and the hormone is can't spell gibberellin. No, 2 B's. 1 R. I think you get some leeway on gibberellin. Who would remember what it's like? It is important that brewers know how the age of their barley affects the concentration of amylase produced, so they can obtain the best yield using the apparatus shown. So that's this apparatus. So we're going to be doing a clear zone thing. Design a method to investigate the effect of age of seed on concentration of amylase. So we're using the clear zone diameter as a measure of the concentration of amylase, and we're altering the age of the barley seeds. So we're going to use a range of different ages of barley grains. Let's have a look and see if we can work out a sort of time scale. So time after sowing, 2, 4, 6, 8, 10. So why can't we use that? So ages, 2 days, 4 days, 6 days, 8 days, 10 days old. Place on starch agar. With the same concentration of agar on each plate. Incubate 24 hours. At the same temperature. So we're getting all our control variables in. Use the same mass of seeds in each experiment. You don't want a big fat one that might have lots of amylase in and little teeny tiny ones. Repeat the investigation and calculate a mean. Diameter of clear zone, let's call that the dependent variable. So I've got my independent, my dependent, how I'm going to do it. So I've got my control variables in, and the only other thing I could do is suggest a control experiment. A suitable control, given that we're looking at enzymes, would be to use boiled barley grains to show the barley is producing the enzyme. Tip top, job's good 'un.